Thanks for joining us. I'm Dr. James Henry, a professor in the Dan F. Smith Department of Chemical Engineering at Lamar University. Today I'm going to discuss how DNA evidence is analyzed. We've all watched movies and TV shows where they catch a criminal or identify a parent from the DNA samples. But how do they do it? Well, according to television, it goes something like this. Some guy in a lab coat puts the sample into some sort of elaborate machine, and we never really see what it does, we just know it's there. And then some magical molecule creature starts translating millions of letters flying past him at the speed of light, carefully dodging as not to interfere. And then, what seems like really only seconds, the letters are all sequenced and read by that same magical machine, and they're compared to some mysterious database. And, if we're lucky, the display eventually pops up the picture of a person the DNA belongs to, and then some scientific expert makes some miraculous declaration. Unfortunately, that's not exactly true. In reality, scientists have to perform numerous laboratory techniques to do this, and they can take days, weeks, or even months. Today we're going to explain one of those techniques with an experiment you can do at home. The technique is called gel electrophoresis. It does have some things in common with what you see on TV. It takes a special machine and computer to read the results that looks something like this. Each of the blue marks in the picture tells us and the computer something about the DNA sample. With enough of these, we hope to identify more information about the source of the sample. This could be species if it's not human, race, gender, or other genetic anomalies. Let me show you how you can do electrophoresis at home. You'll need the following supplies: bottled water, baking soda, salt, food coloring, agar powder, cardboard, aluminum foil, a dropper, 6 9-volt batteries, 2 alligator jumper wires, paper clips, measuring cups and spoons, a microwave-safe bowl, and a small container for the gel. For the next step, you're going to make the buffer that's going to be used for running the gel and making the gel. And to do that, all you need is to take 2.5 cups of bottled water, add a half a teaspoon of baking soda, and mix them until they're thoroughly dissolved. Next, we'll be making the comb. The comb is designed to make holes or wells in the electrophoresis gel to hold our samples. For this, we'll need cardboard, scissors, and aluminum foil. We're going to cut the cardboard into strips that are approximately 1/4 inch wide. This doesn't have to be perfect. Since my cardboard is thin, I'm going to cut twice as many as the number of wells I want, which is 3, but you can do more if you have space. Next, I'm going to wrap the cardboard strips in aluminum foil to make them waterproof. I'm putting 2 strips together in each wrapping because my cardboard is thin, but you can use one if you have thicker cardboard. I do this by placing stacked strips near the edge of the foil and wrapping them tightly. After one layer of foil, I fold it over to seal the end and finally trim the foil. I'll then repeat this 2 more times to create 3 pieces. We now need to construct the comb. To do this, we need to determine how deep the strips should be in the bowl. You want to stop about 1/4 inch from the bottom of the bowl. I use the bowl to measure and mark the strips for placement. We then take a strip of cardboard long enough to bridge the entire top of the bowl and use it to attach the strips to. I'm aligning the marks on the strip to one edge of the bridge piece so that the strip will sit at the depth I want when the comb is assembled and attached. I carefully align all 3 strips to approximately the same length and adhere them to the bridge so that they have a gap between them and the edge of the bowl. Be sure to space them out, and to make this easier, you can use tape, hot glue, really anything to attach them. Notice the gaps that they sit in the bowl without touching the bottom. Then tape the bridge to the bowl to prevent it from moving when you pour the gel to set, which we'll be making very soon. To make the agar gel, take a microwave-safe container. Here I'm using a plastic cup because that's what I have convenient. To this, you're going to add 1 cup of buffer solution that we made earlier. I'm using a 1/2 cup measuring cup, so you'll see me putting 2 in. To the buffer, you're going to add half a teaspoon of agar. It won't dissolve completely at room temperature, and it'll actually kind of start to gum up a little, so you'll need to heat to a simmer in the microwave. Just make sure to watch it so it doesn't boil over, and stir it when it comes out. When it's ready, be sure it's well mixed and pour the solution into the bowl with the comb attached. This will be very hot, so be careful doing this. The liquid should cover at least 1/4 inch of the comb strips, not the bridge piece, and be sure to clean up any messes you happen to make. After pouring, look for bubbles around the comb. If you see them, use a measuring spoon or something else, a piece of cardboard, really anything except your hands, to pop the bubbles so that the top of the gel will be smooth. You need to let it cool to room temperature so that it sets up like gelatin. While it's cooling, we're going to prepare our fake DNA samples for the gel. For this, you'll need some sort of clear syrup. Corn syrup or simple syrup will work. You'll need food coloring also. Then take some of the syrup and add a couple drops of food coloring just to give it a good deep color. You can repeat this for as many colors as you wish to run on your gel. You may be wondering what exactly is agar. Agar, or agar, is a powder isolated from seaweed, and this powder contains a material called agarose. When you heat an agarose solution, the agarose begins to move around and tangle. As you heat more, it moves faster and begins to form coil-like molecular structures. These coils link together to form a gel matrix which, when solidifying, looks very similar to gelatin, forming a jelly-like slab. This slab is what we're going to use to separate our fake DNA samples into pieces. The question is, how does all this work? Imagine this is the gel we are creating with the wells in it. First, we're going to put our samples into the separate wells. For this example, let's assume that the molecules in the samples decrease in size from green to red as shown. Let's also assume that the green and blue are positively charged, while the red and yellow are negatively charged. We can use electricity to create a positive charge at one end of the gel and a negative charge at the other. Based on these charges, and those of the molecules, they'll move toward one end or the other. Based on the size of the molecules, they'll migrate faster or slower, resulting in the separation of the different pieces. Now that the agar has cooled to room temperature and set up into a solid gel, we need to prepare the gel to run. To do this, we first need to remove the comb from the gel carefully as not to rip or damage the gel. We do this by removing the tape holding the comb to the container and gently wiggle the comb while lifting it from the gel. We're going to use a dropper to load our samples into the wells in the gel. But before we do, we need to cover the gel in buffer, making sure to fill the wells and create about a 1/8 to 1/4 inch layer of buffer on top of the gel. Next, we need to cut slots in the gel to hold the electrodes we're going to make. We do this by cutting slots into it, but not through the gel. These slots should be close to the ends of the gel, parallel to the wells in the middle of the gel. To make electrodes, we're going to bend 2 paper clips into L shapes. I was able to do this by hand, but you can use a pair of pliers if that's easier for you. The electrode should be bent such that the long pieces of the L will slide into the slots that we previously cut in the gel, while the short pieces will extend above the surface of the buffer. This is important to allow the proper path for the current to flow. Next, we need to assemble the batteries. 5 or 6 batteries will be assembled in a zigzag pattern, with the small terminal of one battery being inserted into the large terminal of the next battery, and so on as I'm demonstrating. Next, we need to load the gel. We're going to get a little bit of sample in the dropper. Using your other hand to steady it, guide the dropper tip below the surface of the buffer and just to the top of one of the wells in the agar gel. Gently squeeze the bulb of the dropper to begin filling the well. Each well will be filled approximately half full with different samples. Finally, we'll run the gel. We do this by attaching the remaining 2 posts of our battery assembly to the exposed electrode wires using the alligator jumpers. I'm connecting the red to positive and black to negative, which is the normal way for wiring. It doesn't matter which wire is attached to which electrode, since we put the samples in the middle of the gel. Once you connect to them, you'll notice bubbles start forming at the electrodes almost immediately. This is perfectly normal and indicates the system is conducting electricity. I'm going to secure the wires with some tape just to make sure they don't move. Finally, verify everything is in position properly, and now we wait for about 2 to 2.5 hours, and I'll speed this up for you guys. After the gel is run for a few hours, we disconnect it and see what we have. You'll notice the electrode connected to the negative side seems to be fine, maybe a little darker. However, the positive one is rusted due to the electron flow leading to oxidation. But what we are really interested in is the gel. You'll notice the yellow moved the furthest, then orange, green, red, and finally blue. This is due to charge and size of the molecules that make the food coloring dyes. This is very similar to what you would expect if you were running different sized pieces of DNA. Hopefully this gave you some insight into how DNA is analyzed in crime labs. I hope you feel inspired to try this yourself. You can let your electrophoresis run longer or use a couple more batteries to see how far and how fast the different colors move. I hope you've enjoyed this first installment of the Lamar Learning Lab webinar series. Look for announcements of more videos and live events, and have a great day.